Hello friends, in the previous videos, we have seen the overview of eukaryotic DNA replication. We also compared prokaryotic and eukaryotic DNA replication. You can refer my videos, the link is given in the description box. In this video, we are going to discuss about initiation step of eukaryotic DNA replication. Mechanism of eukaryotic DNA replication is similar as prokaryotic DNA replication, but certain differences are there. This we have also discussed earlier. First is larger size of DNA compared to prokaryotes. Hence, multiple origin of replication is present in order to finish the replication on time. Linear DNA. Hence, end structures are present called telomere. Therefore, end replication is required, which is done by telomerase. This we will discuss in detail in separate video. DNA packaging distinctively involves histone proteins. So we already know that in eukaryotes, DNA is a coiled structure. This we will discuss in detail in separate video. Because of these complications, DNA replication is complicated and it involves several enzymes and other proteins. We already know that DNA replication has three steps: initiation, elongation and termination. Here we will discuss about initiation step. Initiation, it is to start or to initiate. It needs to be regulated because of multiple origin of replication. Hence, replication is initiated at many sites simultaneously. So, for each part of genome to be replicated exactly once per cell cycle, tight control is required. If it is unregulated or there is any error in the regulation, then it leads to over-replication that causes DNA damage and genomic instability. This may be due to the repeated initiation events at the same origin. To prevent this, replication initiation is divided into two steps. First is licensing, loading of inactive precursors of MCM2 to 7 helicase. Second is firing, that is activation of the replicative helicase, and these occur in different phases of cell cycle. Licensing occurs in G1 phase and firing occurs in S phase. These two steps are interconnected. Replication begins at origin, which has conserved DNA sequences. In yeast, it is known as ARS, autonomously replicating sequence. Now let us see different events that takes place during the initiation in detail. ORC, that is origin recognition complex, binds to the origin of replication. This then recruits CDC6 protein, cell division cycle 6 protein. After that, CDT1 and MCM227, which is hexamer, bind the ORC CDC6 complex. CDT1 is chromatin licensing and DNA replication factor 1. This CDT1 stabilizes single MCM227 hexamer and loads onto the chromosome. Hence, what we say that MCM227 is loaded. This MCM227 forms the core of the DNA replicative helicase. Both ORC and CDC6 are bound to ATP. So, loading of MCM227 triggers ATP hydrolysis and this releases CDC6 and CDT1. Now what happens? Loading of another MCM27 takes place. Same steps are repeated and CDC6, ORC and CDT1 are released. Now two MCM27 hexamer are present. This forms double hexamer. This double hexamer encircles double-stranded DNA. Thus pre-replication complex is formed. So this is the origin of replication. ORC comes and binds to it. Then it recruits CDC6 protein. Now CDT1 and MCM27 comes and bind to this ORC CDC6 complex. MCM27 is loaded by CDT1 And this loading of MCM227 triggers ATP hydrolysis, as a result of which CDC6 and CDT1 is released. Now there are ORC and MCM227 present. Again the process is repeated. Again ORC recruits another CDC6 protein, and then CDT1 and MCM comes and binds to this ORC CDC6 complex. Same process is repeated. MCM227 is loaded and CDC6, ORC and CDT1 is released. Now this is double hexamer present at the origin, and these encircles double-stranded DNA. This is known as pre-replication complex. So all the above mentioned events takes place in the licensing step of the initiation, and these events occurs in the G1 phase of the cell cycle. As the transition of cell cycle takes place from the G1 to S phase, level of kinase protein DDK, that is DBF4 dependent kinase, and SCDK, that is S phase cyclin dependent kinase, increases. This DDK then phosphorylates MCM227 complex, which then allows recruitment of SLD3 and CDC45 protein. Now this SCDK promotes the formation of preloading complex. This preloading complex is composed of SLD2, DPB11, GINS and DNA polymerase epsilon. Here CDC45 and GINS, these are helicase coactivator. This is the first helicase coactivator and this is the second helicase coactivator. Now this SCDK, that is S-phase cyclin dependent kinase, phosphorylates SLD2 and SLD3 protein. This allows preloading complex to attach. This delivers GINS in combination with DNA polymerase epsilon to nascent replication complex. Now GINS associates with CDC45 and MCM227 to form CMG complex. CDC45, MCM227 and GINS complex. This is functional replicative helicase. SLD2, SLD3 and DPB11 dissociates. Hence, double hexamer, which we discussed earlier, is now converted into 2-CMG complex. This double hexamer encircles double-stranded DNA, and as it is converted into 2-CMG complex, then it functions as the helicase. That means it is functional replicative helicase, as a result of which unwinding of the double strand of the DNA takes place by breaking the hydrogen bond between the base pairs. Hence, this CMG complex now encircles single-stranded DNA. And through GINS, DNA polymerase epsilon interacts directly with CMG complex. MCM10, which is associated with the replication origin, then facilitates the recruitment of RPA, which binds to the single strand of DNA and stabilizes it and prevents its re-annealing or rewinding. It also facilitates the recruitment of DNA polymerase alpha with the help of CTF4. This DNA polymerase alpha synthesizes RNA primer less than 10 nucleotides long, which is then followed by the synthesis of DNA stretch less than 15 nucleotides long. So this DNA polymerase alpha has 4 subunits. 2 subunits function as the primase and synthesizes RNA primer, and the other 2 subunits function as the DNA polymerase and synthesizes DNA stretch less than 15 nucleotide long. Then DNA polymerase delta is loaded. Leading and lagging strand synthesis takes place by DNA polymerase epsilon and DNA polymerase delta. So DNA polymerase epsilon synthesizes leading strand and DNA polymerase delta synthesizes lagging strand. But it can be vice versa because it is not fully clear and research is still going on. So when the transition of the cell cycle takes place from the G1 to the S phase, then DDK, that is DB4 dependent kinase protein, and SCDK, S-phase cyclin dependent kinase protein, their level increases. DDK then phosphorylates MCM227 complex or the hexamer. This phosphorylation allows recruitment of CDC45 and SLD3. The CDC45 is helicase co-activator. SCDK promotes formation of preloading complex that includes SLD2, DPB11, GINS and DNA polymerase epsilon. Now what happens? This SCDK phosphorylates SLD2 and SLD3. This phosphorylation allows the attachment of preloading complex, as a result of which now GINS along with DNA polymerase epsilon comes in contact with CDC45 and MCM227 hexamer and forms CMG complex, which means CDC45, MCM227 and GINS. This is the functional replicative helicase. Also DPB11, SLD2 and SLD3 dissociates or these are released. So as the two CMG complex is formed, the double hexamer which encircles double-stranded DNA is now converted into 2 CMG complex. Since it is functional replicative helicase, hence unwinding of the 2 strands of DNA takes place by breaking the hydrogen bond between the bases. Now CMG complex encircles single-stranded DNA. Also MCM10 associates with replication origin, then it recruits RPA and DNA polymerase alpha with the help of CTF4. So here MCM associates with replication origin, then it recruits RPA, which is single strand binding protein which binds with the single strand of the DNA and stabilizes it and prevents its re-annealing or rewinding. It also recruits DNA polymerase alpha with the help of CTF4. And this DNA polymerase alpha has 4 subunits, 2 function as the RNA primer and 2 function as the DNA polymerase. Hence this DNA polymerase alpha synthesizes RNA primer less than 10 nucleotides long, and then it is followed by the synthesis of small stretch of DNA that is less than 15 nucleotides long. Then DNA polymerase delta is loaded and synthesis of leading strand and lagging strand takes place. This we will discuss in detail in elongation step of eukaryotic DNA replication. This is all for today's video. In the next video, we will discuss about elongation step of eukaryotic DNA replication. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.